Hi everyone. My name is Ruth Epstein. I just finished the third year of my PhD at Cornell University in plant brain genetics, and today I'll be talking about my ongoing thesis work that's focused on understanding how the maize recombination landscape was altered during its domestication event. So to begin, recombination is a really important evolutionary process. It acts by facilitating adaptation to the stacking of beneficial alleles and the removal of deleterious ones. It can also positively influence the amount of genetic diversity within populations over time. And it can also influence the efficiency of artificial or natural selection, which acts by breaking up linkages between selected and unselected loci. And a couple studies have highlighted that selection can in turn affect recombination rates. And this often happens when you have intense selection pressure on a recombination modifier locus or loci. And this occurs when the cost of recombination is far outweighed by the benefit of it. And so this possible cost is infertility, incorrect chromosome segregation, the breaking of positive LG, that is linkages we actually want. And then this cost is outweighed by the benefit, which is breaking up linkages between unfavorable loci. And as I mentioned, a few studies have looked at how selection can influence recombination rates. And the first of which was a study in the Evolution Canyon in Israel. And these researchers compared wild strains of a fungi on a south-facing slope, which had really harsh environmental conditions compared to a north-facing slope. And they found that the south-facing slope, due to natural selection, had much higher recombination rates. And a more recent study looked at wild populations of Drosophila in completely different environments and found that natural selection was again influencing global recombination rates and thus had significant differences between them. And these studies demonstrate that really intense selection pressure on the genome can alter recombination events. And a really prominent way to have selection pressure over a really short amount of time is during crop domestication events. And there has been past hypotheses that have suggested during domestication, there's increasing recombination rates. So a study in 2004 set out to understand this link, and there was a meta-analysis conducted on the number of chiasmata per bivalent between wild and domesticated pairs of plant species. And chiasmata are just the visible sites of crossovers that you can quantify under a microscope. So this meta-analysis found that domesticated plant had much higher chiasmata per bivalent, which means it had much higher recombination rates. However, this study looked at a lot of plant species, and as I mentioned, I'm much more interested in maize and its domestication. So maize domestication is a really well-studied event. It took place about 9,000 years ago in Southwest Mexico, and the wild progenitor of maize is teosinte. And from the schematic, you can see it has a much more branched phenotype. It also has a completely different ear morphology from what we consider to be a corn cob. And because this event is really well studied, we know a lot of the traits to be altered during domestication, and we even know some of the underlying genes that cause this radical transformation in phenotype. And a couple years ago, researchers set out to understand the selection pressure on the maize genome during its domestication. So this graph is 18 different traits in maize known to be under domestication. And it's comparing the inferred selection intensity on these traits to just a bunch of traits under natural selection in teosinte, the wild progenitor of maize. As you can see from this graph, these 18 traits that are known to be involved in domestication had much higher selection intensities acting on it. So all of this shows that natural selection or selection in general acting on a population can influence recombination rates. And given that selection pressure across the maize genome maybe wasn't linear wasn't linear due to certain traits being involved in domestication, I wanted to set out if we could understand where in the genome crop domestication, in particular maize domestication, increases recombination rates. And there's only one example of past studies characterizing um, recombination rate evolution and where it's happening in the genome. And that is with PRDM9. So PRDM9 is a zinc finger protein that catalyzes the formation of hotspots in most vertebrates. It's very well studied in humans. And PRDM9 is also called a speciation gene because due to its fast evolving nature, it can lead to hybrid incompatibility between populations down the line. But this schematic shows the differences in PRGM9 zinc fingers between human and chimps. They're completely different. Other studies have also showed that they do not share any recombination hotspot positions, which is also known between maize and teosinte. However, although PRDM9 seems like a great candidate for understanding how recombination rates are evolving, plants lack PRDM9, which means plant recombination landscapes are evolving differently. Um, I was able to get some empirical population data on maize and teosinte because we have a lot of populations of teosinte. And I looked at the recombination landscape of these two, and I found that they had a really similar distribution across chromosomes. However, a maize did have a much higher recombination rate compared to teosinte. I also found that maize and teosinte did differ in their hotspot positions. They did have some overlap, but most of their hotspots were not shared. However, I take this with a grain of salt because this data only allowed for about 60 kb resolution, and because I'm interested in understanding how recombination rates are changing on a really local level, I wanted to use something that had much more finer resolution. And to do that, I have used population genetics approaches to infer past recombination events. And I did this through using ARG inference. So ARGs are ancestral recombination graphs, and they're really cool data structures that contain all recombination and coalescence events in the history of a sample. And ARGs are often generated from coalescence with recombination. And I will demonstrate how ARGs work in this schematic. We have 4 chromosomes, and this is their inferred genealogy. And the dark blue dots are recombination events, which split lineages going forward in time. And the light blue dots are coalescence events, which merge lineages going forward in time. And ARG inference allows us to understand not only where in the genome recombination events took place, but also possible ages of when they happened. So using when and where recombination events happen with ARG inference, I will want to obtain the teosinte recombination landscape at fine resolution. And I was able to use ARG inference using the ARG Weaver software developed by the SIPO lab at Cold Spring Harbor. And I used ArgWeaver on 26 samples of it's from a diverse maize panel. As you can see from the schematic, the ear and plant height is very diverse. And recently, about 2 years ago, all of these samples had their own de novo assemblies created, thus making this analysis and this data really high quality and also possibly allowed for base pair resolution of these events. But before I get into the results, I want to talk about the specific time intervals I'm using to infer teosinte recombination landscape. So as I mentioned, maize was domesticated about 9,000 years ago. So inferred recombination events that occurred less than 9,000 years ago, I infer to be maize, and then recombination events that happened more than 10,000 years ago, but less than 20,000 years ago, I infer to be teosinte. And just as a side note, teosinte is an umbrella term for a couple subspecies of Zea maize. However, maize was domesticated from Parviglumis, and there were some some admixture events after it was domesticated. But I was using these recombination events and their dates. I was able to recreate the maize and teosinte recombination landscapes and thus compare them. So this is a histogram of recombination rates of 10 kb intervals across the maize and teosinte chromosome 1. And from this distribution, you can see maize has much higher recombination rates compared to teosinte. And teosinte also has a slightly different distribution of recombination events, having a slightly skewed right tail. These results provide evidence for past hypotheses that recombination events do increase during crop domestication events, but also this demonstrates that arc inference can be used to possibly infer ancestral recombination landscapes. But as I mentioned, I'm really interested in where in the genome these recombination rates are significantly changing. So I compared maize and teosinte at the 10 kb level. I calculated the fold change between maize and teosinte per 10 kb interval, and I thus took the extremes from the 95% confidence interval. And I then plotted these changes over chromosome 1. So pink dots is where maize recombination rates are increasing significantly compared to teosinte, and the blue dots is where teosinte is significantly increasing its rate compared to maize. And as you can see from this, there's no obvious bias in where recombination rates are changing. However, this could signal that recombination rates are changing on a much more local level than previously thought to have been. But in summary, I've demonstrated that significant selection pressure on the genome can alter recombination. And during crop domestication events, there has been data and hypotheses that this co-occurs with an increase in recombination. I've also demonstrated that arc inference can possibly use to infer past recombination landscapes. And I also have shown more evidence that maize has increased recombination during its domestication, which validates past hypotheses. In the future, I hope to explore regions of the genome with significant changes in recombination rates. And I want to thank the Pulaski Lab, Melissa Hubitz, Adam Siepel, and Armin Shepin, and Cornell Bioinformatics Facility for making this project possible. If any of you have any questions, feel free to email me at rke27@cornell.edu. Thank you.